All right. So we left off talking about T helper cells and how they are activated when they're naive, meaning they haven't encountered any antigen before. Those naive T helper cells are activated by dendritic cells that have taken up pathogens, shredded them into little pieces, and loaded them onto MHC2 and put them on their cell surface for T cells to swing by, or actually for lymph node T cells to encounter. Right, the dendritic cells more so come to the T cell. The T cells are just waiting around in the lymph node, hoping to encounter antigen, right? And then those naive T cells, once they encounter antigen and receive co-stimulation, B7 cytokines, other co-stimulatory signals, those T cells, once they encounter antigen, they proliferate. You make between 100 and 1,000 new T cells from a single T cell that was activated. And they differentiate. So they turn into effector cells. So T helper cells, effector T helper cells, those are short-lived. They turn into memory cells, which are much more longer lived. They may live the entire lifespan of the individual, in fact, but they probably have an average lifespan of like 10 years. And your short-lived effector cells can turn into memory cells as well. So it's not necessarily true that these will all die. They can turn into memory cells. And memory cells, when they encounter antigen again, they turn into effector cells. So these cell types can revert back and forth depending on what the situation is. And also, you're going to get some follicular helper T cells, which is a topic that I'll probably expand upon more. But basically, these are T cells that develop during every kind of immune response. And they are specialized, super specialized to provide B cell help. So they are responsible for a lot of antibody production. That is their job, to stimulate antibody production. Okay. So I got at this idea that there were a bunch of different T helper cells after differentiation. Okay. So depending on what the pathogen is and where it is in the body, you're going to need a different response to hopefully try and kill it. There's bacteria that sit on the gut and destroy gut cells. There are bacteria that invade cells and go inside cells and explode them once they're done growing or whatever, once they've grown to a certain amount. And then there are viruses that grow inside cells. There are parasites that grow inside cells or grow on mucosal surfaces or whatever. And then you've got fungi and all that jazz, right? So with all those different things, there's got to be different ways to take care of them. And the T helper cells are one of the most important ways that an immune response can become polarized. Okay? T helper cells, when they're naive and become stimulated with antigen, they turn into a certain kind of T helper cell. They may turn into a Th1 cell, a Th2 cell, or a number of other Th cells. Okay? So when they're stimulated with antigen, depending on what cytokine they're provided, or what profile of cytokines is present in the microenvironment, which in turn will be influenced by what pathogen there is, it's going to tell the naive T cell what kind of T helper cell to turn into. The first kind of T helper cell is the Th1 cell. And these are cells that promote cell-mediated immunity, which is one of the modules of immune defense. Sometimes you'll hear this called type 1 immunity because it's Th1, right? And Th1 cells, they are super duper important for mounting responses against viruses and anything that replicates inside cells. So if the infection is inside of a cell, then a Th1's response is going to take care of it. What they do to do that is they activate macrophages to kill pathogens that have been taken up. So they're going to provide macrophages with interferon gamma, a cytokine that greatly activates macrophages. And so they turn macrophages into what we call the M1 phenotype, M1 macrophages. These are macrophages that are very good at killing and ingesting things and shredding them to pieces. Okay? They are not promoting homeostasis. They are promoting chaos and death. Okay? quite literally. Th1 cells also activate cytotoxic T cells, CD8 positive T cells, to kill virally infected cells. So they help promote that response. And Th1 cells activate B cells to produce IgG, which can help coat any pathogen that's outside of cells with antibodies so that they can be taken up by macrophages and degraded and shredded. And in addition, there is a time preference for the response polarization. Usually Th1 cells are early. Okay? Their cytokine profile has 4 major cytokines, okay, that you should probably know. Interferon gamma, interleukin 2, tumor necrosis factor, and beta. Okay? We'll talk about the function of each of these in the context of Th1 cells later on. Then there are the Th2 cells, which are sort of the antithesis of Th1 cells. They promote humoral immunity, also called type 2 immunity. And this is the immunity that helps promote responses against extracellular eukaryotic parasites, okay? Things like worms and, extracellular parasites. Doesn't have to be a worm, could be something else too. Also, molds, fungi that are in the mold form during disease. Th2 cells promote defense against extracellular pathogens by activating B cells to produce 3 isotypes of antibody: IgE, IgA, or IgG. They promote class switch to any of these 3 classes, and you're going to get a little bit of each for a type 2 response. Okay, not just one, but all 3 will be present. Th2 cells activate granulocytes so that they will attack eukaryotic parasites with more anger and hatred. So they're going to turn on eosinophils, they're going to turn on mast cells and basophils, so that those cells begin to make even more antiparasitic proteins, and so that they will release those antiparasitic proteins to hopefully kill the parasite. They turn macrophages into the M2 phenotype, which is anti-inflammatory, anti-helminthic, and reparative. Okay? They are typically promoted later into an infection. And this kind of ties in with the whole repair thing. If you want to begin repairing the tissue, you probably want more M2 macrophages, not M1 macrophages, because you don't want macrophages to still be tearing it up in there. You want them to be promoting repair. M2 macrophages make a bucket load of proteins that help promote epithelial cells to regrow and increase the rate of epithelial cell turnover. And M2 macrophages are also capable of making multiple different compounds that are anti-parasitic. Okay, the macrophages are involved in the parasite response as well. I feel like we usually associate them more with like the parasite response, or excuse me, more with like the bacterial response, but they are involved in defense against parasites as well. They're a very versatile cell type. And Th2 cells produce a lot of ILs. They produce IL-4, IL-5, IL-6, 9, 10, and 13. Each of these has a little bit of a different job that it does to help promote the anti-parasitic immune response. But there are more T helper cells, and you will have to know these for immuno. If you're taking micro, I don't really think we cover these as much, but definitely know these for 425. There are the T regulatory cells, and these are cells that don't really promote a response. They suppress the immune response to things. And we need these. We really need these. These are positive cells for maintaining homeostasis so you don't die. Basically what they do, they inhibit the immune response, they inhibit inflammation. They are favored in physiological states. So there is no infection, there's no damage, there's no inflammation present. We don't have an invader. We don't need to have an inflammatory response of any kind. Therefore, Treg cells help inhibit inflammation. They promote tolerance, meaning lack of response, to non-pathogenic microbes that are present on mucosal surfaces. So you have a lot of bacteria, fungi, and even some protozoa in your gut, right? You have a ton of organisms in your gut. We do not want to mount a response against every organism in your gut. That would be bad. A lot of those organisms are not only they're not only not harming you, so it will be a waste of energy to fight them, but they also help you. They prevent infection by just coating the gut and making sure nothing can attach. They compete with potential pathogens for nutrients and such. And a lot of the things in the gut also make vitamins that we need for survival, or that at least help us to survive. For example, the synthesis of vitamin K by certain gut bacteria. So we don't want to target all those bacteria in the gut. So Treg cells are going to respond to peptides derived from commensal bacteria. And in response to the peptides, they're going to release cytokines that turn down inflammation. They also help to prevent autoimmunity against self-antigens. So they are somewhat self-reactive, at least a lot of them are, and they will respond to peptides derived from self-proteins loaded into MHC2. And when they respond to those self-peptides, they're going to make proteins that suppress inflammation and say, okay, we don't need an immune response. All that there is here is self. Okay? When I say self, I mean host proteins, right? Proteins that are not foreign. They aren't from a pathogen. Treg cells help to inhibit an otherwise excessive immune response from causing too much damage to self. So even during an infection, we still need Treg cells, because if we don't have them, we do way too much damage on accident. Okay, so they're going to help limit the intensity of the inflammatory response. They can promote tissue repair after the pathogen has been killed as well. Okay? Their two major cytokine products are interleukin-10 and transforming growth factor beta. Then there are the Th17 cells. They promote microflora defense at mucosal surfaces. And this really shouldn't say microflora, it should say pathogen defense, basically, at mucosal surfaces. This is what we call type 3 immunity. Even though it's not Th3 cells, this is type 3 immunity. Okay. Th17 cells, you can see them as being sort of the opposite of Treg cells. There are a lot of Treg cells at mucosal surfaces that are helping to discriminate between non-harmful and harmful bacteria. And you also have Th17 cells that are responsive to harmful bacteria and will promote inflammation in response to them. So Th17 cells, they are favored in inflammatory states that would be seen during infection or damage. They're really good at promoting defense against extracellular bacteria and fungi, especially at mucosal surfaces like your barriers, your gut, your intestinal epithelium, and your skin. Lots of Th17 cells there, and in your lungs, etc. Th17 cells promote production of chemokines by the epithelium, which results in the recruitment of neutrophils and other effector cells. They also produce cytokines that cause the production of antimicrobial proteins by epithelial cells. So some innate defense proteins are going to be greatly upregulated when Th17 cells kick in. And they prevent pathogenic and opportunistic mucosal flora from invading and damaging sterile tissues. So you can think of them as sort of being the first line of defense in terms of T cells against mucosal pathogens. Okay. They are especially important in the event of a breach in the epithelium. So if you sort of have a little ulcer or something like that, you better hope for a Th17 response. Our two major cytokine products are IL-17 and IL-22. And we'll talk about the functions of all these cytokines in the next PowerPoint. And then we've got a couple of minor ones that aren't as important, but still know of them. There's the follicular helper T cells, and these are CD4 positive T cells. Their job is to provide help to B cells. They promote class switch, proliferation, and differentiation of B cells. Okay, so they're important for the antibody response. They produce IL-21, which is just a general B cell activating cytokine, and they also produce type-specific cytokines. So in the event of a promotion of the type 1 response, like a Th1 response, they're going to be making interferon gamma. In the event of a Th2 response, they'll be making IL-4. In the event of a type 3 response, they'll be making IL-17. So they're going to make IL-21 and then one of the other type-specific cytokines. probably more than one other type specific cytokine. But they sort of mirror the response that's being promoted. Okay, but you're going to get some of these regardless of what response there is. If it's a Th1 response, you'll be making TFH cells that are making IL-21 and interferon gamma. If you're promoting a Th2 response, the TFH cells are making IL-21 plus IL-4 and probably some other Th2 associated cytokines like IL-10. Yeah. And then we have Th3 cells, which is sort of a dead area of research. No one cares about these anymore. It never really took off, but they're thought to suppress inflammation at mucosal surfaces. They are induced after you eat something that doesn't cause any kind of damage. That's part of their like peptide response thing, their repertoire. So they can be induced after the administration of an innocuous oral antigen. They are not a well understood population. Research into these cells is really never took off, but they produce transforming growth factor beta and IL-10. They are distinct from Treg cells, though. I'm not exactly sure how, but they have different surface antigens. And then there's the Th9 cells, which in general promote parasitic defense. They promote mast cell activity, Th2 differentiation, and anti-parasitic immunity. And they produce IL-9, which helps to drive further Th2 differentiation. This cell type is not important, in my opinion, but Dr. Sanders cares a lot about this IL-9 thing. So know about IL-9 and how it can drive Th2 differentiation. And you can kind of throw the rest of this out of your mind because it doesn't matter as much. Although you may want to know that it also acts on mast cells and helps recruit and activate mast cells. So, as a summary, Th0 cells, naive T helper cells, they are activated by dendritic cells presenting antigen in the lymph node, and then they proliferate and differentiate into effector T helper cells, which then circulate, enter the infected tissue, and provide help to other immune cells in the form of lymphokines, which is just another word for a cytokine made by a lymphocyte. Oops, I have a repeat. Some of your activated T cells become memory cells, which have a much longer half-life. It is possible for a T cell to immediately become a memory T cell upon antigen presentation, or an effector T cell can also become a memory T cell. And some of the T cells that you get from an activation event become follicular helper T cells. They stay in the lymph node and they help B cells. We'll talk more about B cells in a bit. Okay, and then there are the cytotoxic T cells, which are CD8 positive T cells, sometimes called CTLs, for cytotoxic T lymphocytes. These are cells that actually kill other cells that are infected. Okay, these are the only true direct killers of the adaptive immune response. Cytotoxic T cells, they have a T cell receptor. That's what makes them a T cell. They have CD8 as a co-receptor, and that makes them specific for MHC1, not MHC2, but MHC1. They also express CD28, especially when they're naive. They need B7. They need co-stimulation in order to become activated. Okay. CTLs, their job is to target virally infected cells, and when they find a virally infected cell that has MHC1 with a viral peptide loaded into it on the surface, they say, hey, you are infected with a virus that I know to have caused trouble for a dendritic cell that activated me way back in the lymph node. Therefore, you are infected with this virus, so I'm going to have to kill you. And they're going to make very similar proteins to execute killing to natural killer cells. They make granzymes like granzyme B, perforin, and granulysin. So they're going to induce apoptosis very efficiently, and then they're going to move on and survey the next cell to see if it's virally infected, and so on. Okay, they're going to survey all of the cells in the tissue to see if they are virally infected. CTLs also express a relatively important protein called Fas ligand, which binds to a receptor called Fas to induce apoptosis. I'm not 100% sure on the significance of this, actually. I know it can help with the killing response, but it also has like questionable activity in like regulation of the immune response. So you can kind of forget about FAS. It's really not important for this class. The antigen for a cytotoxic T cell is a specific peptide that is typically a foreign peptide that is loaded into MHC1, MHC class 1 molecule. Okay? Like naive T helper cells, your CD4 T helper cells, or Th0 cells, whatever you want to call them, the activation of a naive CD8 positive T cell requires co-stimulation in addition to the T cell receptor becoming stimulated by antigen. Okay? The naive CD8 positive T cells are sometimes called TC cells. The nomenclature can differ from place, so don't quote me on it, but usually if they're naive, they can be called TC cell, and you'll be referring to a naive CD8 cell if you say TC cell. So they require co-stimulation when they're naive. That is signal 2. Signal 1 is the T cell receptor binding to MHC1. The co-stimulation comes from dendritic cell B7. On the cell surface of the dendritic cell, you have B7, which binds to CD28 on the naive cytotoxic T cell. Cytokines are also a good co-stimulatory signal for cytotoxic T cells that are naive. One of the more important ones is IL-2. A lot of times you need a good Th1 response in order to get like an adequate cytotoxic T cell response. So I have a figure showing you that in a moment. Upon binding antigen and receiving co-stimulation, the naive cytotoxic T cell proliferates and it differentiates into effector cytotoxic T cells and memory CD8 positive T cells. Neither of these two cell types requires stimulation. Neither of these two cell types require co-stimulation the next time they encounter. So to become activated in the tissue and kill something, they don't need B7. Okay? So remember that. They only need B7 when they're naive. They don't need it when they're effector cells. Effector CTLs, CD8 positive cytotoxic T lymphocytes, they leave the lymph node where they were just activated. They circulate through the blood, and then they find the inflamed area. Okay? That tells them that there is an infection there. They say, oh, this is inflamed. These vascular endothelial cells have adhesion proteins that I can bind to. I'm going to pull myself out of the blood and into the tissue where there is inflammation. Hopefully, they are entering the tissue where that antigen is from, right? Because they're then, once they get to the tissue, they're going to survey all of the cells for MHC1 with their specific peptide. And they will kill any cell that is expressing their specific peptide antigen inside of an MHC1 molecule. That is a signal that the cell has been infected with the virus. Okay? If it's got MHC1 loaded with a viral peptide on its surface, that is a signal to the CTL that this is a cell that's infected with a virus. Okay? Now, some viruses want to downregulate the CTL response. They want to avoid killing by CTLs. So what they do is they turn down the expression of MHC1 on the surface. And they think, oh, great, now CTLs cannot recognize that I have infected this cell. I can live out here the rest of my days, and nothing's going to happen to me. Well, that is a double-edged sword, because as soon as MHC1 is turned down on the cell surface, natural killer cells become activated, right? Remember, natural killer cells are turned off by MHC1, and if a cell doesn't have MHC1 on the surface, the natural killer cell will kill that cell. So these cytotoxic T cells work together with the natural killer cells to make sure that any cell that's virally infected, regardless of whether it has MHC1 on the surface, it's going to be able to be targeted by either of those two cell types. If there's no MHC1, natural killer cells kill that target cell. If there is MHC1 and it's loaded with viral peptides, which it will be if the cell is virally infected, if it's loaded with viral peptides, then effector CTLs will kill the cell. So regardless of MHC1 expression, you have a cell type that will take care of that infected cell. Okay, the title of this slide is the following: In many cases, naive CD8 positive T cell activation requires the help of CD4 positive T cells. So a lot of times, CD8 positive T cells can't become activated all on their own. They need the input of T helper cells, usually Th1 cells, in order to become activated. Okay, so they're activated in the same place, right? They're activated in the lymph node. That's where naive CD8 positive T cells are. That's also where some T helper cells are, and it's where a lot of naive CD4 T cells are. Now, the reason that having a Th1 cell is important or can help is that T helper cells can provide interleukin 2, which can help promote the growth and the co-stimulation of cytotoxic T cells. T helper cells can provide that IL-2, and T helper cells can also induce the increased expression of co-stimulators on dendritic cells. So if a dendritic cell only has a little bit of B7 on its surface, that's usually not enough to get you a CD8 positive T cell to become activated, a naive CD8 positive T cell to become activated. However, if a Th1 cell swings by and it encounters its antigen inside MHC2 on the surface, it can tell the dendritic cell to begin making a buttload of B7, which can then activate the cytotoxic, naive cytotoxic T cell. And that Th1 cell can provide the IL-2 to help drive more co-stimulation of the cytotoxic T cell. So regardless, actually that's not the right word, but basically Th1 cells can help drive the cytotoxic T cell response through this mechanism. However, in some cases, the dendritic cell has enough B7 on its surface to get you co-stimulation of the CD8 T cell. And in those cases, you don't necessarily need the Th1 help, although it always does, provide a little bit of help for the response. But it's not always 100% needed. Okay, if the dendritic cell is angry enough, if it's really activated, or maybe it's even infected with the virus, then guess what? You can get an adequate CD8 response to it. Okay, but the Th1 response always helps with the CD8 positive T cell response. So this is the slide that shows you how natural killer cells and CD8 positive T cells work together. In scenario A, in both scenarios, you have a virally infected cell. In scenario A, the virus does not downregulate MHC1. Okay, so there's plenty of MHC1 on the surface. Those MHC1 molecules will have been loaded with viral peptides. Okay, so this is a viral peptide, this little triangle here. And a CD8 positive T cell that was activated in the lymph node by a dendritic cell that was, expressing some of those viral peptides in MHC1, it swings by, it circulates, it enters the infected tissue, and it surveys all the cells in that tissue for this molecule here. And when it finds it, it binds to it with its TCR. CD8 binds to the outside of MHC1 to serve as sort of better adhesion between these two molecules. So it stabilizes the TCR to MHC1 connection. That's the job of CD8. And when that happens, when the cytotoxic T cell finds an antigen, finds the viral peptide loaded into MHC1 in tissue cells, it kills those cells that have that MHC1 protein on their surface, as long as it's been loaded with a viral peptide. And now the infected cell dies. This is super duper important for things like viral infection because viruses cannot replicate without a host, right? So if you destroy the reservoirs of infection, and then you also take up all of the virus with a macrophage, ingesting them and then shredding them, well, guess what? The viral infection is resolved, hopefully. Okay. The CD8 T cell disconnects and it goes on about its merry day. It goes and surveys other cells for the MHC1 plus viral peptide. Okay. We're going to look at the structure of MHC molecules a little bit so that maybe this might make more sense about how this whole loading thing looks. On the other hand, in scenario 2, you still have a virally infected cell, but it does not have MHC1 on the surface. The virus has produced a protein or some other molecule that downregulates the expression of MHC1 on the surface. This means that the natural killer cell's KIR, or killer cell inhibitory receptor, is not engaged by binding to MHC1 because there is no MHC1. So there is no inhibitory signal for the natural killer cell. The natural killer cell is going to recognize activating ligands that are present on, almost all cells, and it's going to say, "Oh my God, this cell doesn't have MHC1, and it does have activating ligands. Therefore, I better kill this cell." And it's going to release the same effector molecules as the cytotoxic T cell would have made, but in this case, it's just the natural killer cell making them, and it's going to induce apoptosis of the target cell. These two cell types ensure that regardless of whether MHC1 is on the surface of the target, it's going to be recognized by either the cytotoxic T cell or the natural killer cell. So they work together to provide a full range. And they use pretty much the same effector molecules to kill. Now here's a little bit on the CD4, CD8 thing to clarify. This is something you really need to know really well because I use these words a lot and they're on the exam. They're all over the exam. Okay. T helper cells express CD4. That's why we call them CD4 positive T cells. This means that they recognize MHC class II molecules. So T helper cells are responsive to MHC class II molecules. Part of the reason why they respond to MHC2 and not MHC1 is because they have CD4, which binds to the outside of MHC class II to help stabilize the T cell receptor to MHC class II connection. Okay, so what you don't see in this drawing, you don't see the T cell receptor, and you also don't see the peptide that should be here. But I'm sure you can imagine those two things are present here. This is just to show you that CD4 actually binds to MHC2, and that helps stabilize the connection between the T cell receptor and the MHC molecule. Okay, this is a very important slide to commit to memory. On the other hand, oh, and by the way, MHC2 only expressed on professional antigen presenting cells. On the other hand, cytotoxic T lymphocytes express CD8, which is not even related to CD4. The structures are very different. Okay, they're not even in the same family. CD8 binds to the outside of MHC1, MHC class 1. And that helps to stabilize the TCR to MHC1 connection during recognition of peptide, of antigen. MHC1 is expressed on all nucleated cells and platelets. The only cell that doesn't express MHC1 is red blood cells. Okay? That's the only cell that doesn't have MHC1. But red blood cells aren't exactly a good host for any kind of pathogen. They don't even have DNA. They're kind of just sacks of hemoglobin. There are a number of parasites that infect them, but you really don't need MHC1 expressed on red blood cells. So again, you don't see the T cell receptor here, and you don't see the peptide, but there will be a peptide during this event. Okay. The way to remember this is that CD times MHC equals 8. So if it has CD4, it recognizes class 2 MHC because 4 times 2 equals 8. If it has CD8, it recognizes class 1 because 8 times 1 equals 8. Okay, that's one way to remember it. Or you can be like me and just study this so much that it's like the back of your hand. Either one works. So that was T cells. And I haven't heard any questions or anything yet, so I'm going to assume that there are none, or you're just so frantically writing down notes that you'd rather not. But are there any questions? Okay. So B cells then. These are our last real major cell type. Okay. B cells are adaptive immune cells, and they are lymphocytes. They produce immunoglobulins, which are also called antibodies. And antibodies bind to specific antigens and enable effector functions. One of those functions is they mark bacteria or whatever for uptake and destruction by macrophages. Now, there's a lot of different functions that they enable, though. We'll talk about those functions, slideshow 4. B cells are actually antigen presenting cells. They actually present antigen. So they express a B cell receptor. And that B cell receptor, basically just an immunoglobulin, it's just an antibody molecule with a little membrane domain attached on the end of it. And the B cell receptor binds to an antigen, and then it triggers the uptake of that antigen into the B cell. The B cell then shreds the antigen inside that endosome that it was taken up into, shreds it into little pieces, and it loads the peptides onto MHC2 molecules, puts them out on the cell surface, and then it looks for a nearby T helper cell to present it to. The goal of this process is the B cell wants the T cell to provide it with cytokines. And those cytokines are important activators of B cells. Okay, things like IL-6, IL-21, pretty much any cytokine can act on a B cell. They express a lot of receptors for a lot of cytokines, but they're looking for T cell help. Okay, that is why they're antigen presenting cells. One super important thing to remember about B cells, they can bind to like anything. Their antigen is not MHC. Their antigen is not MHC. Their antigen can literally be any molecule. As long as it's foreign, right? If it's not foreign, you might have an autoimmune disease get triggered. As long as it's foreign, a B cell can respond to it. So virtually any structure can be recognized by B cells. And taken up, shredded, loaded into MHC2, presented to T helper cells, and you receive T cell help, helps you get an antibody response. The B cell receptor on a naive B cell, that is a B cell that has never encountered antigen before, it's just waiting to encounter its antigen. The B cell receptor on naive B cells is IgM. Okay, it is always IgM. It is a membrane-bound molecule of IgM. The binding of the B cell receptor to an antigen triggers the uptake of that antigen, shredding of the antigen, the loading of the peptide pieces of the antigen onto MHC2, and the upregulation of B7 on the surface of the B cell. It's actually called B7 because it was first found on B cells. The B cell then moves to the nearest lymph node T helper cell to present the antigen. It wants to receive T cell help. When a B cell presents antigen to a T helper cell, it causes the T cell to secrete lymphokines or cytokines that provide the B cell with T cell help. It's called T cell help. Whenever a T helper cell responds to antigen with cytokines, we call it T cell help. Those cytokines help drive the response to the infectious agent. In this case, the T cell help is helping the B cell undergo class switch and helping the B cell to secrete antibodies. So one job of the cytokines is they tell the B cell what antibody isotype to make. This is a process called class switch when your original isotype changes. You're always starting with IgM when it's naive. Sometimes when the T cell provides cytokines to the B cell, it triggers the B cell to begin making a different type. The four major types of antibody are IgM, IgG, IgE, and IgA. It's only class switch when you're switching from one class to another, okay? And you can switch from IgM to IgG to IgE, for example. You're not locked into the class that you switch to. You can't go backwards, though, and we'll talk about why that is way later. But you can't go backwards. You can only go in this direction. IgD is technically also a class of antibody, but it has like sort of an unknown function, and it's not secreted. It never becomes secreted as an antibody molecule. After a B cell receives those T cell cytokines, it receives T cell help. It then proliferates. So you go from one B cell to like 100, maybe 1,000 B cells. And it differentiates into plasmablasts and then into plasma cells. Those plasma cells are really what to remember. They crank out a bunch of soluble antibody proteins that have a single specificity and a single isotype. For example, a single plasma cell may be making antibodies. It's going to be making antibodies. And this particular plasma cell, it is making IgG antibodies that recognize a very specific structure. Okay, it's only going to bind to one thing and one thing only. And that's all that it's going to make, is that one amino acid sequence of antibody. Different plasma cells make different antibodies that have different specificities, and sometimes they have different classes as well. There's only 4 classes, but there is an unlimited number of specificities that an antibody molecule can have. Okay. Memory B cells are also a thing. So when you activate a B cell, when a T cell activates a B cell, you also get a memory B cell. These memory B cells do not need T cell help the next time around in order to turn into plasma cell. They just hang out in the lymph nodes and in the tissues, and when they encounter the pathogen's antigen again, they rapidly turn into a plasma cell and begin making a crap load of antibodies. Those antibodies help to bind to the pathogen and mark it for destruction, etc. Okay, that's how B cells work. Still no questions so far as that's okay. Process this at your own rate. So this is a slide that really explains everything in one paragraph. B cells, they encounter antigen using their B cell receptor, and they receive co-stimulation. So they actually have to receive co-stimulation, and for B cells, that comes in the form of complement fragments. We'll talk more about that later. After they encounter antigen, they take it up through receptor-mediated endocytosis. They shred the contents of the endosome into little bite-sized pieces. They load any resulting peptides onto MHC2 molecules and put them on their surface. MHC2 has been loaded with peptide. It's put out on the surface for a T cell to swing by and recognize. B7 is also expressed to help give the T helper cell some much needed co-stimulation. Then they present the peptides to a T helper cell. In response to the MHC2 with the peptide binding to the TCR, the T cell receptor, the T cell responds by providing the B cell with cytokines. We're talking pretty much anything can be a cytokine. Ooh, there's a typo here. But anyway, those cytokines are going to act on the B cell to do something, and it's probably going to promote multiple things. It's going to drive proliferation of the B cell, class switch to a potentially different class of antibody, somatic hypermutation, a process we'll talk about later, and a terminal differentiation event into a plasma cell, which is a short-lived cell that makes a ton of antibody. They are basically antibody factories. They make tons of antibody. They only make antibody of a single amino acid sequence. It's going to have one specific. It's going to have one antigen it can bind to, and it's going to be of one isotype. Okay, but it is tons of antibody. Note that the T helper cell does not recognize the same antigen as the B cell. The T helper cell responds to MHC2, loaded with a specific peptide. That is the antigen for a T helper cell. A B cell's antigen can be any molecule, literally any molecule, with the exception that it has to be foreign. Okay, it usually can't be a self molecule. It has to be foreign. Otherwise, you're looking at autoimmunity. Okay. This slide is actually not something I even really need to talk about. Just demonstrates the concept that T cells recognize different portions of molecules than B cells, and that's totally okay. Not a slide I need to talk about, though. So, we have now introduced, well, technically 4 types of antigen-presenting cell, right? We've introduced the dendritic cell. We've introduced the macrophages and monocytes, which are pretty similar cell types. One is in the tissue, one is in the blood. And then we also just introduced the B cell. All of these express MHC2, but they do it for very different reasons. Dendritic cells express MHC2 so that they can activate naive T cells. That is the whole point of a dendritic cell presenting antigen. They move to lymph nodes where they can activate naive T cells. They also express very high levels of B7 so that they can get adequate co-stimulation to T cells. And then we have the macrophages and monocytes. Macrophages and monocytes, when they present antigen, they're not trying to activate a naive T cell. They want to present antigen to an effector T cell, T helper cell, an effector T helper cell. And when they present antigen, they want T cell help. Okay? They want those juicy, yummy cytokines to activate them and make them super good killers. They're trying to take care of the pathogen and kill it. B cells, on the other hand, when they present antigen, it is derived from things that they've taken up through their B cell receptor, as opposed to phagocytosis. They take them up through the B cell receptor. They shred it into pieces, load it onto MHC2, present it to T cells. They're also looking for cytokine help, but they're not looking for cytokine help so that they can kill things. They're looking for cytokine help so that they can begin to make a bunch of antibody. They want to undergo class switch and turn into plasma cells that dump tons and tons of antibody into the bloodstream. Those antibodies then diffuse into tissues where they coat pathogens and mark them for destruction. Okay, B cells, they're looking to make antibody. Macrophages, they're looking to be killers. Dendritic cells, they're trying to activate naive T cells to kick off the adaptive immune response. One final point. Only dendritic cells can do this thing called cross-presentation. This is when a dendritic cell takes up a pathogen or some kind of molecule from a pathogen, and it can load those molecules into MHC1 despite the fact that it was taken up from the external environment. They are the only cell that can do that. Most cells, when they take something up from the environment, they can only load it onto MHC2. And to load something onto MHC1, it has to have come from inside the cell. In dendritic cells, they can actually take things up and load them onto MHC1. That is called cross-presentation. Okay? And that is an extremely important for the activation of naive CD8 positive T cells. Okay? They're the only cells that can do that. And that makes them good at activating both kinds of T cells. Okay? So here's a table comparing and contrasting all of the major cell types that you probably want to know. This table can help sort of organize this very large amount of material that there has already been. And I apologize, but this is not exactly an easy topic, as you can probably tell. But this table might be helpful for you to look at, just to remind yourself. If you know everything on this table, you're probably good for the most part. These are the subsets of cells, how many there are in the blood, and their lifespan. So, I don't know, sort of interesting. And this is a table showing you the differentiation of all blood cells. Anything that comes from bone marrow precursors. Most of the stem cells that give rise to the immune cells and the blood cells, they are in the bone marrow, the red bone marrow. Okay, and this is just differentiation pathways. But really, what I want you to realize from this is a few things. There are lymphoid cells, which are lymphocytes. They give rise to lymphocytes, right? And then there are myeloid cells, and those give rise to everything else. So your lymphocytes are 3 types of cells: B cells, T cells, and innate lymphoid cells, which includes the natural killer cells. Ignore lymphoid dendritic cells. Those are a weird exception. So really, there's only 3 types of cells that are lymphocytes. Everything else is a myeloid cell. Okay, but within the myeloid cells, you have the granulocytes, which are basophils, neutrophils, eosinophils, and mast cells. And then you have the mononucleated cells. These are your monocytes, macrophages, and dendritic cells. And then also from the myeloid progenitor, you have technically platelets and red blood cells, but we don't care as much about those in this class. 2 other things. New T cells develop in the thymus. That is where T cells are made. New B cells develop in a special area in the bone marrow. We'll talk about T cell and B cell development in agonizing detail for exam 2. So this is a sort of summary of the different kinds of pathogens that can cause you to die, or just can cause you to be, less evolutionarily fit for survival. Um, there are pathogens that exist in the interstitial space, blood and lymph. Those are taken care of with different mechanisms than those on epithelial surfaces. Then there are cytoplasmic parasites and pathogens, and vesicular pathogens. And all of these require a slightly different response. And then this table also compares and contrasts, I guess, sort of the ideal bacteria, viruses, fungi, parasites, and intracellular bacteria, and the mechanisms by which they are dealt with. So different pathogens, different responses is the point of this slide. And then there is this topic called the mononucleated phagocyte system, or the reticuloendothelial system. So basically, it's just a bunch of macrophages that are located in different tissues. They're all given different names. This slide is not for your memorization's sake. This slide is for your clarification's sake. Okay. Macrophages that are located in different tissues are given different names, and they comprise the mononuclear phagocyte system, or MPS, which used to be called the reticuloendothelial system, or RES. In the skin, there are cells called Langerhans cells. Those are macrophages of the skin. There are macrophages in granulomas. We'll talk about granulomas later. There are macrophages in the lungs. We call those alveolar macrophages. And then the rest of these aren't as important, but if you hear their names mentioned, it just might be good to know the names in case you get confused, basically. So, yeah. And then this is a figure showing you all that jazz. This is another figure showing you all that jazz. So this is all the different macrophages. And it also turns out that there's two major sources of macrophages. There are macrophages that come from the blood monocytes, and then there are the macrophages that are self-renewing in the tissues. And those actually are from the early yolk sac during the development of the fetus. The yolk sac makes these macrophages that diffuse to the tissues, and then those are there for their life, for your whole life, basically. And then we've got some esoteric cell types. These are good for you to know more later into the course, but just telling you about them up front is also a good idea. So B2 B cells are the standard B cells. There's also B1 B cells, which are kind of weird. They have more restraints on their functioning than B1 cells, but they can pull off more interesting things. These are the cells that give you memory B cells. These are the cells that can do somatic hypermutation. They can undergo lots of clutch without too much restriction, etc. They are produced only after birth in the bone marrow, and they include follicular B2 cells and marginal zone B2 cells. The follicular cells, those are the ones that we've pretty much been talking about this whole time. They make antigen-specific antibodies during an adaptive immune response. And they are mostly living in the lymph nodes, in these structures called the follicles, where they await antigen. But they also circulate from lymph node to lymph node, and from blood to lymph node, and from lymph node to blood. They're constantly looking for antigen. Okay. Marginal zone B cells, B2 cells, they are in the marginal zone of the spleen, but they are B2 cells. They are a first line of defense against blood-borne pathogens, and they are fixed in the marginal zone of the spleen. So they're actually like tethered to the tissue there. They are suited to recognize and respond to blood-borne bacterial cell wall antigens, stuff like capsules and such. More frequently than not, they do not need T-cell help in order to begin secreting antibodies. But that's all fine and dandy. The only issue is they can't really class switch. They don't give you memory cells, and they are not boostable. So if you vaccinate multiple times, these cells, the response from them does not increase. Okay, so they're more limited in what they can do, but they also do not need T-cell help. So they're a much more rapid way of making antibodies. And if you have a bloodstream infection, those are generally pretty rapidly deadly. So you can see the utility of having a rapid antibody response against something circulating in your blood that shouldn't be there. Okay. And then we have the B1 cells. B1 cells, there's actually two more kinds of B1 cells, B1A and B1B. They're produced by their B cell receptors being stimulated and the cytokine IL-5 or the toll-like receptors binding to pattern recognition receptors or binding to PAMPs. And they secrete what is called natural antibodies. These are antibodies that are made before you're born all the way up until when you die. Natural antibodies, they have low affinity for many antigens, including common bacterial polysaccharides, but they also bind to fixed antibodies that are, bound to an antigen. So they help to just coat things that are already an issue. They help just amplify the marking of that issue. And they bind to intracellular self-antigens. So this function is important because natural antibodies help clear out apoptotic bodies from cells that have died. They help macrophages recognize little apoptotic bodies and take them up and degrade them, basically. They clean out the tissue of dead cells. B1 cells cannot undergo class switch. They're restricted to IgM, which again, that is the first class of antibody on B cells. They cannot give you memory. And it actually turns out that antibodies made by a B1 cell can have specificities for a variety of antigens. That is not to say that they're making a multitude of different antibodies. It's just that they happen to make usually IgM molecules that are capable of recognizing a lot of related structures with low affinity, but that's okay because it still helps. Because of their reduced affinity, these natural antibodies, they usually require complement to become activated in order to opsonize effectively. Lots of terminology here, but it'll make more sense later when we talk about complement and such, and antibody. They're first made in the fetal liver. The B1 cells are first made in the fetal liver, and then they self-renew in the periphery after birth. They're not so much in the lymph nodes, but they hang out in the peritoneal and pleural cavities. Although they are also technically found in lymph nodes in the spleen. So this is a table comparing and contrasting all of those different types of B cells. And there are several types of T cells. There's the alpha beta T cells. Those are the T cells that we have talked about so far. They make cytokines if they're CD4 positive, or they kill cells if they're CD8 positive. But we also have these weird T cells called the gamma delta T cells. They have many distinct populations that are, they emerge during different times during fetal development, and they go to different tissues and they make different cytokines, and they also respond to different antigens depending on when they were made. They express a genetically distinct, usually invariant T cell receptor. So these gamma delta T cells, they frequently recognize many of the same antigens, and they are involved in a multitude of processes. They don't respond to MHC proteins, but they may respond to other antigen presentation molecules that are a little bit strange. CD1 proteins that have been loaded with lipids. They usually don't express CD8 or CD4, but they do express CD3, which tells us that they are indeed a T cell subset. Oh, did I cut out? Can you guys hear me? Can you guys hear me?
Yes, I'm trying to leave the session, but I'm not able to. I don't know what I'm doing.
Oh, okay. Let me end the recording. We'll just stop here for today. It's